All right, let's get into it. Today, we're not just going to answer a GCSE biology question. Oh no, we're going to absolutely master it. We're going to take a real question, use the official AQA mark scheme, and I'm going to show you how to nail every single mark.
So, here's how we're going to do this. First, we'll break down the question. Then we'll hit the core knowledge parts, look at the potato experiment, crunch some numbers, and draw a graph. And finally, we'll tackle that big osmosis explanation. Let's go.
Right, Section 1. So this whole question is all about cells and transport. That should immediately get you thinking about, cell parts, how things get in and out, stuff like osmosis. That's the ballpark we're playing in.
Okay, let's move on. We're diving into the first few questions now. This is all about that core biology knowledge, the stuff you just have to know. These are the easy marks we want to lock in right away.
And here we go, our first question. It's a classic table filling exercise. You've probably seen a ton of these. 3 empty spots, 3 marks just sitting there for the taking. This is a straight-up test of your memory on cell parts and what they do. Okay, so here are the perfect answers, straight from the horse's mouth, the official mark scheme. So what contains the genetic info? That's the nucleus. And mitochondria? Their job is respiration. Now see those brackets around site of aerobic? That's a little hint from the examiner. It means just writing respiration is totally fine. And the last one, what controls what goes in and out? That's the cell membrane.
But wait, there's more. This is where the real magic of using a mark scheme comes in. Look at this. For nucleus, you could have also said chromosomes and still got the mark. For respiration, releases energy is good to go. But, and this is a big but, you cannot say it produces or creates energy. No way! That's a classic mistake. Energy is released, not made from scratch. It's a tiny difference in wording, but it's the difference between a point and 0.
All right, next question. One simple point for the function of chloroplasts. Sounds easy, doesn't it? Well, it is, but there are a few ways to get the mark, and one really common trap that people fall into.
The golden answer here is, of course, photosynthesis. That's the one you're aiming for, but what if you said something else? Let's check out what the mark scheme says.
And here's that trap I was talking about. You see that last point? The examiner will ignore contains chlorophyll. Why? Because that's what a chloroplast is, not what it does. It's not its function. You have to say it absorbs light or produces glucose. You've got to link it to the actual job it's doing.
Okay, this next one is a bit of a clever one. It's not just recall, it's about applying what you know. So if chloroplasts are all about photosynthesis, and photosynthesis needs light, what part of a potato plant is never going to see the sun?
The first thing that probably pops into your head is a root hair cell, right? And that's a perfect answer. But check this out. The mark scheme gives you a whole list of other correct answers. Xylem, phloem. Knowing these can be a real lifesaver if your mind goes blank on the day. All right, changing gears now. We're moving away from pure recall and into a practical investigation. And it's a famous one, the potato and salt solution experiment. You've probably done this in class. So here's the step-by-step method that was used. Now, every single one of these steps is important, but the examiner is about to zoom in on one of them to check if you really understand why you do it. So, question 01.5 asks, why on earth did the student dry the potato pieces? What's the point of that step? Think about it. What could go wrong if they didn't? And this slide is so important. A lot of people would just write, to remove water from the surface. That sounds right, but it won't get you the mark. Look at the full mark answer. You have to explain the consequence. You have to say that if you didn't dry it, the mass would be higher, which would mess up the results. It's all about linking your action to the result. Okay, next up, the numbers game. We're getting into calculating and graphing. For this section, you've got to be super precise. Every little detail matters if you want to scoop up all the marks. First, a 2-marked calculation. Now remember the rule for these, show your work. You almost always get 1 mark just for the method and the second one for the right answer. So let's walk through it. Step 1, find the increase in mass. Easy enough. 2.7 minus 2.5 is 0.2 grams. Boom, that's 1 mark right there. Step 2, plug that into the percentage change formula. So that's 0.2 divided by the original 2.5 all times 100. And that gives you your final answer, 8%. That's your second mark. Done. And now for the main event of this section, the 4-mark graph. I know, graphs can look a bit scary, but just think of it as 4 separate 1-mark jobs. One for the scale,
One for the axis label, one for plotting the points, and one for the line. We can do that.
Okay, let's look at the examiner's notes. They are super picky about this, so listen up. Your axis label needs the name and the units. Your scale has to be big. It needs to use at least half the grid. And the big one, your line of best fit has to be a curve. If you just connect the dots with a ruler, you're going to lose that mark.
All right, now you have to use the graph you just drew to answer this question. It's asking for the concentration where the potato's mass didn't change at all. So where on your graph is the percentage change 0? You're looking for where your beautiful curve crosses that horizontal x-axis. Now, obviously, your answer will depend a little on how you drew your curve, but check this out. The mark scheme is actually pretty nice here. Even if you completely messed up your line of best fit, as long as you give an answer somewhere between 0.31 and 0.33, they'll still give you the mark. That's a good safety net to know about.
And here we are, the final part, the big 3-mark explanation. This is where you have to tie everything together and explain the science behind what happened. Why did the potato lose mass when it was in a really salty solution?
Here's the recipe for a perfect 3-mark answer. It's like a checklist. Point 1, you have to say that water moved out of the potato. Point 2, you have to name the process. What is it? It's osmosis. And point 3, you have to explain why. It's because the solution inside the potato was less concentrated or had a higher water potential than the solution outside. Hit those 3 points, and you've got your 3 marks. It's that simple.
So there you have it. You can see that knowing the science is just the start, right? The real secret is understanding what the examiner wants to see right down to the exact words. That's how you turn a decent grade into a top one. So, looking back, which one of those little mark scheme details surprised you the most?